This is a capstone project partnered with Violumas, who manufactures UV LED. Here you can see the picture of a prototype UV LED provided by Violumas. The aim is to test the efficacy of UV LED device in disinfecting grocery packaging in retail outlets to prevent microbial spread. As we all know that the effective disinfection of respiratory droplets on fomites and surfaces are very important to prevent the spread of infectious diseases. Surfaces like floor, bench tops are usually decontaminated with chemicals, but chemicals are not suitable for food and its packaging material as they produce toxic effects due to its chemical residue. The current pandemic of COVID-19 pressed us to think about the effective disinfection method for these materials. An alternative to chemical, UV is a well-known method used to disinfect water and surfaces for decades, but conventional UV lamp contain hazardous mercury bulb. To eliminate these hazards, UV light emitting diodes is an emerging technology needs to be evaluated for its effectiveness. So we studied the effectiveness of UV LED in disinfecting food packaging materials on which very limited study was conducted. We selected Staphylococcus aureus and E. coli as bacterial representative and MS2 as a surrogate marker of virus. We used overnight culture of E. coli, S. aureus and MS2 suspension for spiking aluminium foil surfaces. Then it was UV irradiated at 265 nanometer wavelength for 1 and 2 minutes. UV treated and untreated foil pieces were transferred to phosphate buffered saline and a tenfold serial dilution was done. We used spread plate culture for E. coli and S. aureus, and from each dilution, 100 microliter was transferred on blood agar plate for colonies. For MS2, we used pore plate culture in E. coli K12 bacterial host in soft agar lawn using standard double layer plaque assay for each dilution. The result of our study showed 4 log reduction of bacterial growth in UV LED treated plates. This is a real picture from our laboratory, and you can see there is no growth in all our 1 and 2 minute UV LED treated plates compared with untreated. We observed 2 log reduction in plaques produced by MS2 in UV LED treated plates. From our result, we can conclude that UV LED is a hazard-free, effective method of disinfection which is sufficient to disinfect grocery packaging within a minute while moving on the conveyor belt. Because of its smaller size, rapid disinfection, easy operation and safety, it is promising in food industry and healthcare.